Well, thank you, Maya. Thank you for inviting me to this neuroscience meeting and, in fact, launching a pain whole-day discussion. It is something I've been studying for multiple decades, and obviously it has lots of implications about society. And thank you for the honor of being the speaker in memorial to your dear colleague and scientist who you sent me a paper that he has published in 1968 about the size of the observable universe that he theorized. I'm sorry, I don't know the theory, using Hubble equations, etc. But I checked on Google that the number he proposed in 1968 still remains essentially intact, which is amazing. So I wish I would have done something along those lines. Anyways, I want to talk about pain in general, and I want to talk about and sort of give you an overview of what we have been doing and where we are going in the field in general. It's more of a discussion rather than a lecture, and I want to sort of show you the directions that we're going in overall. I don't know, does this work? No, how do I advance this?
Right now.
It dings instead. Wait, am I doing the wrong thing? Oh, okay, all right, sorry. Sorry, okay. And before I actually discussed our science, I want to at least mention that, you know, my work is embedded in a larger community of scientists, and this is a center that we have been working within over the last 5 years or so. In fact, some of the people who you will hear about from tomorrow were members of this center to start with. Overall, this is a center, fully translational center, where we do both human and animal studies, going back and forth between data that we collect in both species, hoping to create a scientific basis with which we can address the huge societal need for treating chronic pain. Before I move to the topic of chronic pain, I want to first start with pain in general and pain from a standard viewpoint of, you know, you injure your finger, and obviously there's some local injury that's painful. You push on a pin, or you burn your finger, and obviously you have activated some nociceptors on the skin that transmit that information to your brain, and we feel pain. On the other hand, what I want to also make the strong claim that pain and nociception should not be confused with each other. In general, when you have an acute painful stimulus, obviously nociceptive activity is involved. On the other hand, nociceptive activity is present for all the time as we exist and does not give rise to pain perception, in a sense that all species, all motile species have nociceptive responses to the environment, but we don't think the amoeba has pain perception, but they do have nociceptive reflexes. So overall, my simple claim, although I don't have the evidence for it, is that nociceptive activity is an unconscious signaling for behavioral modification. In a sense, when you are sitting in here, this is my little joke that I have to share with you, you are fidgeting a little bit, maybe because you don't like what I'm saying. My simple alternative explanation is that your nociceptors are protecting you behind from generating injury while you're not feeling any pain. And in a sense, from that viewpoint, pain is not a protective signal, but is a signal of the failure of the nociceptive signals protecting the organism. And as a result, in a sense, pain is a conscious signal for the failure of nociceptors, which then demands revoking new behaviors, new learning, etc., that will change the behavior so that we will not burn the finger again, right? So I'm going to talk about brain imaging and my favorite technology that we've been using for more than 30 years now, and how brain imaging informs us about perception in general and for pain specifically. Very simply, I don't want to complicate the story. Brain imaging provides us information about the structure of the brain, the axonal properties of the brain, information transfer between areas of the brain, and activity within the brain. So we see which brain areas are talking with each other, how the brain is active, and both axonal and gray matter integrity of the brain. And again, before I move to chronic pain, I want to make this very simple statement. What is our level of evidence from brain imaging data about specificity of brain areas involved in pain perception, acute pain perception, simple injuries or simple thermal stimuli that we can put on the skin and feel ouch and what happens in the brain, versus, for example, visual perception, looking at each other or the brain activity for one or the other. So beautifully, there is now a very simple meta-analysis tools freely available on the internet that you can simply put the word vision in there, and based on 14,000 brain imaging studies, it returns to us the brain activity pattern associated with it. So this is for the vision. Neurosynth is the website. If I put the word vision in there, I get all of these red areas activated, right? On the other hand, if I put the word pain, I get these other areas activated. And note that there's essentially no overlap between them, right? And in both cases, 10% of the cortex is activated, 10 unique regions of the brain. And if I look at the peak activity, it gives me a value, a confidence level of 10 to the minus 23 for that region not being activated in that visual task. If I do the same analysis for pain, I get essentially the same number. So in a sense, the brain activity pattern accumulated over the last 20 or 30 years gives us a similar level of confidence of brain regions activated for vision versus pain. But there's a caveat to this. In the case of vision, from the days of Hubel and Wiesel and on, we have massive data that shows that a third of the brain is specifically specialized for analyzing visual information from outside from the environment. In the case of pain, there is yet to be shown a single column in the cortex that's specialized for pain perception. So even though the patterns and the confidence levels are the same, mechanisms underlying the two systems are not the same. Okay? And in fact, our current ideas about what circuitry in the cortex in fact is really related with the perception of pain remains relatively enigmatic. Anyway, given that introduction, I'm going to move on to chronic pain and not bother with acute pain any further. So the next message I want to give to you is that chronic pain is not equal to the persistence of acute pain. Chronic pain is often generated from an acute injury event, yet overall, of people in general who have a similar injury, a small proportion of them end up developing chronic pain. So the injury by itself is not sufficient to understand chronic pain. I'm not going to go through all the evidence we have accumulated over the last, I don't know how many years, too many years to mention, that chronic pain shows distinct brain properties, distinct types of chronic pain, show distinct reorganization of different regions of the brain. But what I want to emphasize today is that chronic pain is primarily predicted from brain limbic properties. And I will elaborate on that in the rest of my lecture. But importantly, both brain predispositions and reorganization of the brain are fundamental components of chronic pain. So coming back to the slide of brain imaging, what I want to emphasize, again, in general, is that if we look at the brain in chronic pain patients, we see abnormalities about the brain anatomy, about the brain glial integrity, about axonal integrity, about brain activity and connectivity. All of those things are shifted and change with chronic pain, and they seem to change in different patterns, different extents, depending on the type of chronic pain that one looks at. So it's a complicated process, but on the other hand, I think is a viewpoint that's giving us new insights and new pathways with which we can address the problem. Let me skip this. Am I missing a slide? I am missing a slide. The scale of the problem of chronic pain is huge, right? Across the whole planet now, the data shows that chronic pain is one of the leading health issues worldwide. About 20% of the world population reports chronic pain. This is true both in the Western world, in the Southern Hemisphere, across the planet, and the number continuously increases. At the same time, treatments for chronic pain remain essentially non-existent. We don't have any scientifically validated treatments for chronic pain. We use medications that have been validated for acute pain as a means of controlling chronic pain, but we know the evidence has repeatedly shown that those things don't work. Especially in the US, a very large proportion of chronic pain patients are treated with opiates, right? And at least in the US, nowadays, there's 100,000 people dying from opiate overdose. And a large proportion of that, in fact, comes directly from the use of opiates in the medical arena of trying to control chronic pain. So, okay, in fact, my primary question here that I want to address along these lines, why bother with prediction? Why do I want to talk about prediction? The title of my talk is Predicting Chronic Pain. And my simple idea is that if I can predict the future of an event, I'm assuming I am honing into some process that has something to do with the underlying mechanisms, and that prediction can have both clinical utility in decision-making in the clinical arena, as well as in mechanistically concepts that we can use to identify circuits, molecules with which we can develop novel therapies. Let me move on. So, very simply, the standard viewpoint for many, many years has been that you have an injury, that injury persists, and that's a chronic pain state, in a sense that the nociceptive signal from the site of the injury, when I have back pain, what do? I massage my back, or I put some cream on my back, or some non-specific procedure of the sort, trying to alleviate the pain. What I want to show you is that a large proportion of that chronic pain is based on brain properties. So that, yes, the nociceptive signal may be an inciting event, but it is not the event that's persisting over the long term. So, in contrast to this peripheralist viewpoint, what I want to overall show you the evidence is that brain emotional circuitry define a risk for developing chronic pain, and the interaction between this risk and the injury creates this transitional state where subjects either over time recover from it or persist. And in a sense, the emotional learning processes underlie this transition state. And once the condition persists, then the brain in fact, literally develops a new state of chronic pain with its unique properties, unique anatomy, and unique physiology. So that's the overall kind of message of my talk. The rest of it are details, and we can skip the rest of my talk. Okay, let me move on. So again, very simplistically, I don't want to belabor all the brain circuitry and all the details of what goes to which region. If we very generally subdivide the brain into two pieces, the cortex and the subcortex, right? And very simplistically, I'm going to say the cortex is where the conscious perception is happening, while the subcortex is where the emotional learning is happening. In a sense, the subcortex is providing the coloring of the cortex perceptual state, right? And essentially, the interaction between these two is defining both the risk factor as well as the consequences of developing chronic pain. This is another way of illustrating what I will try to show. First of all, again, let me emphasize that nociception is happening all the time. There is nociceptive input every time I take a step, every time I move around. The reason I will not extend my arm more than a given level is because my nociceptors stop me from doing it. So that's a very personal protection of my behavior. Nociception is happening all the time, and it's invading both the limbic brain, the emotional part of my brain, as well as my consciousness. Now, when there's an injury, obviously that signal is much larger. I have a perception of pain. And the long-term responses of this system is where we think the development of chronic pain is happening. If I have the ability from the cortex to inhibit my emotional responses to that stimulus, my pain in time will go away and become a healthy subject. In contrast, if I cannot do that, then the circuitry the emotional circuitry will send signals to the cortex, reorganize the cortex into a new chronic pain state. So that is my, dynamic cartoon of developing chronic pain. Overall, again, we think of it as having 4 different stages. Risk factors, predisposition. The risk factor, together with the interaction with the injury, gives us this transitional state from which we either recover or we end up into chronic pain. Let me just skip on this slide here's some data, in fact, very old data that I want to show you of one of our earliest studies of brain activity. Again, this is the acute pain brain activity pattern that I am using this meta-analytic tool. This is an older slide, but the results are the same. Now, if we instead bring in chronic back pain patients in this study, and look at brain activity for their back pain without any external stimulus, when they are actually rating how much pain they have on their back, the brain activity we found, this was again one of our first studies, is in the frontal cortex, a region that's not in this network, so that in fact the pain for chronic pain, the specific region of the brain that's activated is different from that of acute pain. And in fact, and we can do this in a more sophisticated way. We can look at the spontaneous pain for brain activity in this frontal cortex. The intensity of that spontaneous pain nicely correlates with that activity of that region, while for acute pain, that region is not activated, but another area of the brain is activated. So there's a double dissociation between brain areas involved with acute pain from that of chronic pain. So here's, again, this general concept that if I bring in subjects at an early time for an initial injury and look at their brain properties over time, so if these are the subjects that I bring in, some of them in time will develop chronic pain, others will not. Now, if I can look at their brain properties at time 0, then I can identify risk factors for chronic pain. Okay, and here is a study that we did more than 10 years ago now. This is, in fact, the pain of these subjects. So they all start at time 0 with about the same amount of pain. These are acute, subacute back pain patients. Over time, half of them continuously decrease their pain. This is over a 1-year period, right? While the other half have exactly the same pain, constant over the whole year. So these subjects, we call them recovering, and these are persisting. In a sense, these subjects are developing chronic pain, while these guys are healing properly. And about 50% of the population within this group does each one of them. And a very simple measure, simply the amount of information shared between two brain areas, between, again, this limbic brain area and the frontal cortex. Just this link between these two areas actually predicts long-term outcome from time 0, from the first time we scan their brain up to 1 year later, who is going to be in the chronic pain state versus who is not. And in fact, that gives us a fairly good prediction. We can, for example, 1 year later, we have 80% correct prediction of who is developing chronic pain. And the data and this prediction can be validated in out-of-sample data set. Now, that's a very simple model. We have extended that into a more complicated model. But the important part here is that brain limbic properties, amygdala, accumbens, regions, and their interaction predicts about 60% of the variance of who's going to develop chronic pain or not. And so that's all fine. Then the question is, how can we validate such studies in a cross-species analysis? And in a very simple sense, the area of the brain that we initially first identified, nucleus accumbens, is an area that's well known in addiction literature. Right? It has to do many cocaine, smoking, sex addiction, eating addiction. All of them engage the dopamine circuitry within nucleus accumbens, and so. Then we simply say, if we look at that circuitry in chronic pain, can we identify circuit adaptations now causally in animal models where we can induce a chronic pain-like model and look at local circuit properties. And I'm just going to show you just a few slides. Basically, within a few days after a peripheral nerve injury that persists in pain behavior for the rest of the life of the animal, we see brain activity changes in these dopamine neurons within nucleus accumbens, anatomical changes, structural changes, the dendritic profile of these neurons change, the synaptic properties change, and in fact, the level of dopamine within that region is also changing as well. So, and in fact, if we use optogenetic or chemogenetic tools with which we can change the excitability of these neurons locally, and that excitability, in fact, translates into behavioral changes in these animals. So we can either increase their pain behavior or decrease their pain behavior very simply by changing the level of activity of these neurons. Therefore, I would conclude that these neurons are causally linked to the pain behavior that these animals have. Right? These specific population of neurons. And on the other hand, Okay, sorry, this slide is, I think, is a bit out of order, but it is an important message that I want to say, is that, yes, we can identify that specific population of neurons, but there is an abundance of network within the limbic system that we can, in fact, use to control chronic pain behavior. We can do that from ventral tegmental area, from the nucleus accumbens, from the hippocampus, and from the medial prefrontal cortex. So these circuits are highly interactive, and any one of them can, in fact, modulate pain behavior. And so the circuitry is complex and it's not trivial. So, and the other brain area that I thought I'll give you a few slides about that we think is important in chronic pain is the hippocampus. This is, in fact, work that Dr. Radulovic and I started many years ago, and it continues to give us, in fact, really beautiful results. It is the simple concept that chronic pain is not a pure perception, but it is a memory trace as well, right? And that memory trace is causally linked with the behavior that we see. Let me skip this slide. It's too complicated, and I don't want to bore you with it. But in general, the hippocampal connectivity to the brain is itself predictive of the long-term outcome of chronic pain. And in fact, the hippocampal structure itself is changing over time in people who are developing chronic pain, and it is changing more specifically in females than males. What that exactly means, I don't know, but the specificity is really exciting to know. And more recent data shows that if I change the excitability of hippocampal neurons, I can reverse chronic pain-like behavior in animal models, in rodent models. These are two different models. In each case, we can completely block neuropathic pain behavior by changing the level of excitability of neurons in the dorsal hippocampus. And we can also block the emotional component, the affective component, versus the somatic component of the pain also. Now, very recent studies that I want to share with you is a collaboration with multiple labs. This is data in MTBI, an issue that we've discussed here back and forth. In this case, these are subjects. Again, these are patients that have been studied in Israel, actually. They were scanned within 48 hours from having a car accident. These are all patients that had no chronic pain beforehand, so they're healthy subjects. And we have followed these patients over 1 year. So 250 patients were included in the study. They all had a brain scan within 48 hours after a car accident and no brain injuries, right? Yet signs of TBI. And 1 year later, 50% of them have chronic pain. So let me show you that data. So some 110 people completed the study. This is the pain of those who are recovering. Over 1 year, their pain goes down to 0, while the other group, the other 50%, their pain is essentially unchanged over that year. Okay? Now, if we look at the hippocampal functional connectivity, within the first 48 hours, that there is a strong hippocampal cortical connectivity and time, duration, and anxiety, a three-way interaction that, again, very robustly predicts 1 year later who is going to have chronic pain or not. So this, in fact, is a bit different from my original model. In this case, these subjects, at least at this level, they don't have an obvious risk factor. Right? These are healthy subjects. Okay, we don't know if they don't have any risk factor, but at least this hippocampal involvement here is an acute response to that injury, and the response of the hippocampus and the level of the anxiety of the subject that that they come with, between the two of them, identify, again, who is going to have chronic pain. Another example of another condition that, again, we've been studying, and I just want to sort of go over some of the data, is a population of knee osteoarthritis patients. Again, a very prevalent condition for chronic pain. A huge portion of the population suffers from knee osteoarthritis. Its rate only goes up with aging. And the best treatment for it remains total knee replacement, right? Knee replacement works in a very large number of these patients. About 70% of them have no pain after the surgery. On the other hand, about 30% of them, in fact, have pain essentially the same level. And now, again, we say, okay, if we look at these patients, can I predict who in the future will have pain versus not? Again, not from the surgery, prior to the surgery, from their brain properties. Okay? And if I couldn't, I would not show you the data. And so this is so we have two different studies in this data set, and I want to be transparent and say the two different studies have given us contradictory results. In both cases, we can predict the outcome, and in both cases, we predict the outcome from multiple, again, limbic structures, from the accumbens, hippocampus, amygdala, size and shape changes. One data set comes to us from Spain, from Portugal. The other one is from Chicago. And in one case, the decrease in brain structure predicts outcome. In the other case, the increase. I don't understand it, but it is where this discrepancy is coming from is an exciting venue to explore. Again, limbic properties are still predicting long-term outcome in both of these groups. Let me go back. So let me just show you a few slides of some very recent data we have been collecting. It is quite amazing that even in Europe nowadays, a large number of chronic pain patients are treated with opiates. And I don't know in Slovenia, but certainly in Germany and England and France, in fact, the trend has increased rather than decreased. In the US, the number is in the millions. Yet, to my knowledge, there is not a single systematic study of what happens to the brain of patients who are using opiates for the long term, specifically who are in chronic pain. So this is a study we've been doing for the last 5 years or so, and these are some of the latest data that I wanted to share with you. This is a study now of 70 chronic back pain patients who are on steady dose of opiates, who have been using opiates for their pain relief on average for 6 years. Okay? So the range of opiate use is from 6 months to, I think, more than 20 years. The control group here is chronic pain patients who are matched for their intensity of pain, for their duration of pain, and many other things, for sex, alcohol use, smoking, etc. And now we simply look at both behavioral properties and brain properties. And in fact, if we just give them a bunch of questionnaires, all of these yellow scores are worse in the patients who are on opiates. Okay? Even though their pain is the same amount, their duration of pain is the same amount. So many of the outcomes that have to do with pain-related characteristics have become worse in these patients. Let me just skip all of this just for the sake of time. And in fact, the effects are both sex-dependent and race-dependent as well. The details are complicated, and I'm not going to belabor them. If we look at the anatomy of the brain, again, the patients who are on opiates versus not, the overall whole brain matter volume is decreased in the ones who are on opiates. And in fact, there are very on top of that, there are specific brain areas that are also decreased in gray matter density. That includes some of the sensory motor cortex and the anterior cingulate cortex. More importantly, what happens to brain activity in these patients? Now, we simply look at spontaneous brain activity. I'm not going to belabor the technology. And if we look at the brain activity, you can see that there's a massive upregulation of the limbic brain in activity and downregulation of lateral frontal cortex and temporal pole. And we could, actually approximate what these things are. But again, by and large, this reward value, motivation, learning circuitry are massively upregulated in these patients. And it is independent of the dose and of the dose of opiates they're using, of the dose of opiate in their blood. In fact, in a subsample of this population, when we look at them, when there is no more opiates in their blood, the pattern is not changing at all. So this is a long-term reorganization of brain activity. And again, if we look at patients who actually taper down from their opiate use, and again, a very small group, we see this region of the brain basically decreasing in its activity. So again, the limbic brain is a massive driver of the condition. And again, there is both a sex and race dependence of this pattern as well. So I'm going to conclude that brain characteristics determine propensity for chronic pain. Therefore, we think of it as a neurological vulnerability. So people come with a risk of developing chronic pain or not, and depending on that risk, the same injury may or may not give rise to chronic pain. And limbic circuits are the main risk factors. And these limbic circuit properties and their changes do give us pathways and molecular targets with which we are in fact trying to develop new drugs that are specifically designed for these chronic pain conditions. And the correspondence between humans and animals gives us a whole variety of tools with which we can start exploring those things. So overall, my one-line statement would be chronic pain is a brain being addicted to nociception, but driven by emotional learning processes. And I will stop there. Thank you.